Welcome to San Diego and welcome to ISAP's 24th meeting. Our sponsors, speakers, and meeting organizers have done a great job pulling this together. I hope you enjoy it. If you could all find a seat, get some food if you want. We'll be ready to start. There are a few of the paper schedules. If you want them, you can probably get one, but don't forget we also have apps that you can use to see what's going on today. And with that, our meeting organizers, Peter and Hugo, do you want to outline what we're going to be doing? We'll get started a little bit early since this meeting sometimes runs a little bit late.
Good morning. The micro was not really. Can you hear me okay? Because I think, I'm not that important, but all the speakers are. Are we cool? Yes, we are. Okay, so the two of us were the program chairs, and it was a pleasure, and we tried to really come up with a program that is broader, not just sort of down to purely clinical pharmacology, but as a first program point, we thought, why not start with our core business, clinical pharmacology in anesthesiology, and this is our first session. The second session, because we understand there's a lot of exciting new drug development after kind of a hiatus of a decade or two. And we have several really interesting presentations for you that focus on innovation and entrepreneurship, first-hand stories from someone taken, from just an idea to really a drug candidate that's currently in phase 1 and phase 2 studies. Then we have a lunch and business meeting around 12. After that, followed by a poster discussion, so please all attend. As you know, especially for the young investigators, it's really important, their first poster presentations, that they have an audience and not just talk to the moderators. Session 3 is, we're going to have hear an exciting story from Gary Pelt from Stanford. I'm not going to say anything else. It's truly exciting. Rod Eckenhoff will then give us an update in 1 hour, condense a 3-day meeting, the MEC meeting in Bonn, what's new in anesthetic mechanism, what is what has developed over the last, let's say, 5 years, what we know about how anesthetics act. And then the last session will be a Q&A, an interactive session with the A&A editorial board, Steve Schaefer, Jean-François Pité, and our anesthetic pharmacology editors. And this should be a session that's very interactive about sort of where is the journal, where's the journal headed, lessons learned for authors, for reviewers. And at the last but not least, we're going to hand out the Lifetime Achievement Award. This is always one of the nicest and nicest sessions of our meeting. And after that, we have a reception. So without further ado, may I invite our first speaker, Kent Johnson, to give us an update on clinical pharmacology. Thank you.
Thank you. I'm going to do a little bit of a sound check. Can you guys hear me with this one? Is this one better? Or is this one better? This one's better? Well, I'd like to thank the meeting organizers for the invitation to join you today. Get this guy going here. As mentioned, my topic is what's new in the anesthetic drug pipeline, and I'd like to start off with an acknowledgement. Several people in the audience and others I went to and sought advice and input on this, to which I am thankful. Try to call them out as I go along. The first drug I'd like to briefly talk on is alfaxolone. As you know, it's a GABA-A receptor agonist and a similar chemical structure to progesterone. It has a long history. It was combined with another agent in an excipient cremophore, and it was used in many countries for over a decade. A couple of features that it was noticed to have: it had a rapid onset and offset. It wasn't irritating to blood vessels, had minor cardiovascular and respiratory depression with a wide margin of safety. The problem: hypersensitivity to cremophore. Caused bronchospasm, hypotension, essentially activation of complement. And so it was withdrawn in 1984. Fast forward to current day, Colin Goodchild and his colleagues have taken a cyclodextran and reformulated it. The cyclodextran enjoys low toxicity and a hypersensitivity profile, essentially doing away with the problems of the prior preparation. And so the cyclodextran plus alfaxolone equals a new product, Faxan, otherwise known as Fax. This month in Anesthesia and Analgesia, the results of a phase 1 clinical trial were published, and I'd just like to go over some of those results. In this phase 1, they had 12 subjects in each group, and they dosed it to be equipotent, meaning a BIS less than 50. And they looked at important side effect profiles. Of note, airway obstruction, pain and discomfort on injection, involuntary movements, emergence delirium using the Richmond Agitation and Sedation Scale, and nausea. Key findings: 9 out of 12 folks had airway obstruction. Means during the 30 seconds that they were dosed to a BIS of 50, somebody had to do a chin lift. None for alfaxolone. Pain and injection, again, 8 out of 12 versus 0. And the others were less impressive. Another feature of this study shows that the hemodynamics between the two, again, remember, they're dosed to get an equipotent effect. That's shown here with the BIS data. Blue's propofol, red's alfaxolone. They looked at systolic, diastolic, and heart rate. And probably the best figure to look at here is the systolic blood pressure remains above a mean of 90 versus the propofol, which dips. So it seems to be a sparing of the cardiovascular depression that we're all familiar with propofol. I recently received an email from the author of that paper, the lead author, that wanted to point out some other features that may be of interest with this drug. And this was directed at neuroprotection. And this particular drug also, I hope I can pronounce this right, has an agonist effect on pregnane X receptor, or PXR, in the central nervous system. And just to orient you to that senses foreign toxic substances, and with that upregulates detoxification and clearance proteins, such as microenzyme 3A4 or conjugating enzymes. So the bottom line is that alfaxolone may have both GABA and PXR receptor activity, and while having no hormonal activity that its cousins do. With that, he filed a recent patent a couple weeks ago with that technology. He used some supporting data in that. I'd just like to briefly go over that data for you. In a sheep fetal model, late gestation, looking at apoptosis in animals treated with alfaxolone and an antagonist to pregnanolone, they looked at the number of cells that died. And this is with their data. want to draw your attention to these boxes. This is the antagonist, number of cells, death. Here's both drugs combined. Here's the drug. Here's alfaxolone alone. So it seems to have a sparing effect on cell death. They also looked at, by the way, that data came out in 2006, and this is even older data. It came out in 1989. And again, I'm pointing out this is the data that they had in their patent. They looked at adult cats, 8-minute cardiopulmonary arrest, saline versus alfaxolone mixed with alpha-dolone. And they looked at epileptic activity and neurologic deficits. And what they found was that in both instances, the neurologic deficits and the incidence of epilepsy, we got those backwards there, yeah, much better with alfaxolone. So, in summary, alfaxolone, it looks like it's a GABA-A and possibly a PXR receptor agonist. Its phase 1 trial has been completed, has a favorable side effect profile compared to propofol, and it looks like it may have neuroprotective properties. And Dr. Sarner and I were just talking about this last night, and that seems to be a very favorable position for an anesthetic drug to have that feature. Not only is it an anesthetic, but potentially neuroprotective when most of what we use is not. Next drug I'd like to briefly touch base on is remimazolam. It's, as many of you know, in phase 2 clinical trials, and 2 pieces have come out this year since our last meeting, both of them in the endoscopy suite, one for upper and the other for lower GI. Just like to briefly go over what they found. Here's one that was published in April in ANA. 100 patients randomized to get escalating dose or different doses of remimazolam or midazolam. Then they tried to put the scope in, and they assessed the success of the procedure, sedation levels, and the recovery from sedation. And they found that with increasing doses, they were more successful. They eventually got the job done with all of them with giving a little rescue sedation along the way. There were no instances where it required manual ventilation. So that looked promising. This next piece, I couldn't find it in press anywhere. The manuscript is available in its raw, non-edited form from the Gastroenterology Endoscopy Journal. It's very similar in design. Here we have 160 patients randomized either to several doses of remimazolam or of midazolam. This time they added 100 mics of fentanyl and took a look at the same outcomes. This is their data here. And the doses were a little bit heavier, or a little bit more, I think, than the endoscopy upper GI ones, and they were more successful just getting the scope in right away. One of the differences is that they had a loading followed by a maintenance dose. And that was different from their prior study in April. Manual ventilation required? Again, no, for all these doses. So they're successfully able to complete the procedures, and looks like the phase 2 trial data that was completed was successful. And I want to point out that remimazolam enjoys the nice advantage over propofol in that it has a reversal agent. I'd like to now turn to work done by Doug Rain's lab with cyclopropyl methoxycarbonyl etomidate, or CPMM. Sorry, Doug, if I botched that. It's also known as APV 700. It's an etomidate analog. And this lab has been prolific in putting out a lot of work. I'm just going to summarize a few of the recent papers to set up where they're going now. This first one looks at a comparison with CPMM with propofol, and it was published more than a year ago. What we already know is that it's a GABA-A agonist. It has sedative hypnotic properties. And what's new from this paper is they showed that when they compared it to propofol, the loss of riding reflex in tadpoles had similar potency. So the potency was similar. And then they looked at the EEG recovery after prolonged infusions ranging from 5 minutes to 2 hours. And one potential advantage that they found with CPMM was that the effect dissipated faster. They concluded from that CPMM provides more rapid and predictable recovery than propofol, particularly when there's a long infusion. They took that drug and looked at a larger animal model. As we've clarified, it has a faster hypnotic recovery, and it also has less adrenocortical suppression, and they wanted to look at it in a larger species. What they found after a 2-hour infusion was that recovery was faster than propofol, as before. And they also focused on the adrenocortical recovery, and it was also faster. Take a look at these half times here. 215 minutes versus etomidate, long time. And they compared that to propofol, and after 90 minutes, the CPMM and the propofol were similar. So CPPM compared to etomidate, short versus long, similar with propofol at 90 minutes. That was encouraging. So they wanted to take a look at that adrenal suppression issue in a model of sepsis, where it might be more of an issue. They start off with clarifying that septic patients have cardiovascular instability with anesthesia. Etomidate's nice there because it has minimal cardiovascular depression, but it can suppress adrenocortical steroids that restore homeostasis and improve survival. So they were excited to explore CPMM and look at it, see if they had a rapid return of adrenocortical function. In a rat E. coli sepsis model, they compared etomidate, CPMM, to vehicle. What they found, less steroid synthesis suppression with CPMM, less elevated plasma inflammatory cytokines, and lower mortality. Check that out. 40% in the etomidate group, 0 in the CPMM and vehicle groups. As some of you may know, this preparation has been, the medicine company is now working with this drug, and they just announced the results of their phase 1 study. And CPMM is known as APB 700. And just in brief, this phase 1 trial involved 60 subjects. It was a single intravenous ascending dose. They looked at PK and PD and found that its effects were consistent with what was observed in animals. And there were no adverse effects in this phase 1 trial, except for one subject with moderate tachycardia. A little call out to Michel Struz in his lab. Here, they've got 2 posters that describe this phase 1 clinical trial. So at the poster session, I encourage you to go pick their brain and learn more about that trial. So in summary, compared to etomidate, CPMM has a much improved adrenal suppression, or the adrenal suppression is much less, much more manageable, and it's on its way. It's finished its phase 1 clinical trial. Next drug I'd like to talk about, MDA7. This comes to us from Dr. Naguib's lab at Cleveland Clinic, and as you may know, this is a cannabinoid receptor type 2 agonist. And cannabinoid receptors I did a lot of homework in learning about this drug. There's 2 types, CB1 and CB2. CB1 are everywhere, central nervous system and in several other organs. CB2 are immune system, and part of the immune system of interest here is they're in the microglia. So cannabinoid receptors, CB1, CB2, the CB2s are in microglia. Microglia, they have key roles in central nervous system disorders, from neuropathic pain to neurodegenerative diseases. May be a promising therapeutic target. Microglia have different phenotypes depending on where they find themselves and what's going on. M2 is the good kind, promotes neuroprotection, secretes anti-inflammatory cytokines, IL-4, IL-10. M1, this is the pro-inflammatory, this is what you don't want. Pro-inflammatory cytokines, IL-6, attracting chemokines and reactive oxygen and nitrogen species. When looking at neuropathic pain, activated glial cells are in the M1 state, pro-inflammatory state, release neurotoxic factors, self-perpetuate a neurotoxic cycle. CB2 agonism may break this cycle. And here's a schematic prepared by Dr. Naguib, illustrating some of his thoughts on the mechanisms here. The thing I wanted to point out here was the self-perpetuating neurotoxicity, release of neurotoxic factors that go and hurt the nerve, and the nerve releases more activators of the microglia that create more neurotoxic factors. And perhaps one way to arrest this process is to use a CB2 receptor agonist, which lives on microglia cells. Here's some of Dr. Naguib's work, recently published in 2012. A nice model of neuropathic pain is to give rats 4 days worth of the chemotherapeutic agent paclitaxel, and they get sick pretty quickly. And so he compared paclitaxel to getting nothing at all, to being treated with the vehicle, being treated with the CB2 agonist MDA7 for 4 days versus 14 days, et cetera. Very interesting results. Notice here that this gray line right here, this is just the vehicle. So if you don't get paclitaxel, you don't have any pain problems. And this metric here, PAW withdrawal threshold, is a metric where they apply pressure to the paw at varying intensities, and then they wait till it releases, and it's measured in grams, or the weight on the paw. Look what happens when you get paclitaxel. Painful, quick withdrawal. Look what happens when you give it with the vehicle, same story. Paclitaxel with the agonist for 4 days and 4 days. You get some benefit, and then it goes away. If you give the CB2 agonist for 14 days, it practically returns it to a normal condition. They also looked at this in knockout mice with the agonist, just to confirm that it was really a CB2 receptor. And what they found was that if you gave paclitaxel with an intact mouse, that you got the same response, that you developed allodynia, and then you could reverse that with the MDA7. In the knockout mice, not so. Well, what happens if you come to the party late and the patient's hurting and you want to rescue them? And they looked at that, gave them the paclitaxel insult, and then followed a little bit later with the MDA7. And they found that it's not a perfect win, but things get better here. More recent work from their lab, they've looked at spinal gene expression, profiling, and pathways analysis of the CB2 agonist in the spine following a paclitaxel neuropathy. And what they found is that MDA7 reversed it, or induced peripheral, the preventive effect of MDA7 on paclitaxel-induced peripheral allodynia in rats may be associated with genes involved in signal pathways, in central sensitization, microglial activation, and neuroinflammation. Exciting stuff. So neuropathic pain. This preparation may be able to turn down the inflammatory process, so it may be an important tool in our toolbox for that. And please consult with Dr. Naguib to learn more about this. But I believe he has plans to initiate soon a phase 1 clinical trial. Well, here I've just given you a brief update on a few drugs in the anesthetic pipeline. There are many more. I'm happy to report that the pipeline is relatively full. And much of the innovation looks promising. I'd like to switch gears a little bit and talk about a couple of drugs that I've come across in my role as a section editor. The first one I'd like to focus on is Sugammadex. This slide here charts their FDA approval odyssey. What a journey. Started out in January of 2008. Schering-Plough announces new drug application for Sugammadex. Things got stormy. One issue was a hypersensitivity to the drug that slowed down the approval process. Go a little bit forward here. In July of 2013, Merck statement on FDA advisory committee meeting about Sugammadex sodium injection. Looks like they're getting ready to move things along again. They get a complete response letter telling them we still haven't quite figured out this hypersensitivity issue, and we need more. We need to know more. Our society weighed in on this in July of 2013, and specifically what they said, ASA strongly supports the approval of Sugammadex because it will significantly enhance the ability of anesthesiologists to more safely care for patients due to its novel mechanism of action and its speed of reversal of non-depolarizing neuromuscular blockade. Earlier this year, the FDA cancels Merck Sugammadex NDA advisory panel meeting. Again, they wanted to find out more about the hypersensitivity issue. When looking for that, there is a case report in 2011 about an allergy to low-dose Sugammadex. On clinicaltrials.gov, there is a study entitled Evaluate the Incidence of Hypersensitivity After Administration of Sugammadex in Healthy Participants, last revised a month or so ago. Their main outcome measure was how often did people have anaphylaxis, and they looked at placebo, 4 and 16 milligrams per kilogram, and found none along the way. They looked at some other side effects. They looked at nausea. Looks like the higher doses did have more nausea than the placebo. Let's see, they looked at itching and urticaria, and again, those were higher than placebo. So perhaps these things caught the interest of the FDA. Meanwhile, while all this is going on, this drug has been widely used outside the United States, and a lot of investigators have been publishing their work with it. Here's one such piece. Evaluation of surgical conditions during laparoscopic surgery in patients with moderate versus deep neuromuscular block. So the aim of this paper was to say, hey, if we improve surgical conditions with deep neuromuscular blockade, does it help surgery? And they came up with a surgical rating score. I'd like to draw your attention to this because this will help us interpret the next graph. Extremely poor conditions is a 1, 2 poor conditions, 3 acceptable conditions, wide visible laparoscopic field, but muscle contraction movements or both occur regularly, interfering with the surgeon's work. 4, good conditions. There's a wide laparoscopic working field with sporadic muscle contractions, movements, or both. There is no immediate need for additional neuromuscular blocking agents. 5, optimal conditions. There's a wide visible laparoscopic working field without any movement or contractions. No need for additional neuromuscular blocking agents. Here's their data. So we have moderate neuromuscular blockade, train of 4 of 1 to 2. Deep, post-tetanic contraction 1 to 2. So deep versus moderate. Conditions. In both groups, most are 4 or 5. In fact, if you add up the 4 and 5, you get a high percentage of all the folks. The editor's key points on this. Surgical conditions may be affected by the depth of neuromuscular block. This small study assessed conditions rated on a 5-point scale by a single surgeon during retroperitoneal laparoscopic procedures. Surgical conditions were rated significantly better under deep neuromuscular blockade. Another piece came out in ANA. This is in 2014. And it was a similar type study. And they concluded that deep neuromuscular blockade was associated with surgical space conditions that were marginally better than the moderate muscle relaxation during low-pressure laparoscopic cholecystectomy. Well, not to call out Dr. Naguib again, but he published a review article that came out, I believe, this spring. And it looked at whether or not this approach was appropriate. The title was, Laparoscopic Surgery Muscle Relaxants. Is deep block helpful? Well, he points out that in order to execute the deep block and to use Sugammadex, it has significant economic repercussions, and that there's little objective data to support that deep neuromuscular blockade, when compared with less intense block, contributes to, and I put this part, the most important part here in orange, better patient outcome or improves surgical operating conditions. At the crux of the matter is whether neuromuscular blockade is a substantial improvement in conditions during laparoscopic procedures. Or can we get away with moderate block? Do these data support a change in practice? Scales to characterize the operating conditions and space, do they translate into improved real surgical outcomes, reduced surgical time, decreased complication rates, improved patient outcomes, less postoperative pain, shorter recovery room stay, hospital stay, reduced cost? Deep neuromuscular blockade is likely to improve surgical operating space. Reversal with Sugammadex is expensive. How these conditions translate into improvements in patient outcomes is unclear. So we are awaiting more data to help us make that decision. So in summary, Sugammadex, great drug. It's used throughout the world. Still on a long journey for the regulatory approval. Hopefully we'll get there. Data supporting selected proposed uses, at this point I would suggest is inadequate, and I'd encourage us to take a closer look at that. I'd like to spend the last few minutes talking about liposomal bupivacaine. I noticed that Passira has a table outside, and I'm grateful for their support of our meeting. To start off with, where I work, we do a lot of knee surgery, and my orthopedic joint surgical colleagues have gone to injecting Exparel into the knee joint along with a substantial dose of regular bupivacaine. Mix it up together, put it in the knee. As a member of our acute pain service, on rare occasion, I am asked to supplement their block with an adductor canal block or a femoral nerve block, depending on the situation. And this has become a little bit of an issue because we're like, well, how much bupivacaine did you put in the knee, and can I put more in the thigh or groin? Is it safe? So Dr. Schaefer was kind enough to share a few slides with me about this topic. I'd like to go over some of them with you. Looking at the labeling for liposomal bupivacaine, it's labeled for bunionectomy at a dose of 106 milligrams and hemorrhoidectomy at 266 milligrams. This is important. I'd like you to focus on these doses here. In the drug interaction segment, it says, don't mix Exparel with lidocaine. If you're going to administer it, wait 20 minutes before using some other local anesthetic. And if you're going to use bupivacaine immediately before or mix with Exparel, it may impact the kinetics and chemical properties of the drug. Looking a little bit further, the package insert looked at the plasma concentration for a standard dose over 96 hours. And the standard dose is 20 milliliters of 1.3% or 266 milligrams. So this graph is both reassuring and worrisome to me. First, the plasma concentrations get to a height of probably somewhere under 300 nanograms per ml. And to give you an anchor, 3,000 nanograms per ml is around where CNS and cardiac toxicity may live. The one piece that's missing for me here is a metric of variability. By comparison, consider this study. This is plasma bupivacaine concentrations over time with a combined lumbar plexus and sciatic nerve block. Total bupivacaine, they gave a whopper dose, 300 milligrams, 150 milligrams in each block. They noticed that the peak at about 30 minutes was 1.7 micrograms per ml, or 1,700 nanograms per ml, with a slow decline over the next 4 hours. Some patients did achieve plasma concentrations above 3. That's the toxic level. 300 milligrams hefty dose. That's 60 milliliters of 0.5%. None of the patients developed signs of toxicity. So here's what I like about this study. It's all over the place. This is probably more reflective of what's going on with patients. You got some people that have low concentrations, some people that, whoa, get above the toxic threshold, but it gives me a better feel for what's going on with the drug. So to look at the data that were used for, I'm assuming that were used for the labeling. Here's a piece that looks at comparing Exparel to placebo, and the placebo is saline. And they look at a cumulative over time pain assessment over here. So a placebo is black and the Exparel is the gray. And here is their area under the curve for the first 24 hours in terms of perceived pain control, pain. And that is they found that the bupivacaine was better than saline. My immediate reaction to that is, why not compare it to another local anesthetic? Next study, this is for the hemorrhoidectomy. Management with liposomal bupivacaine. I'd just like to show you one piece from that. So they tracked its the amount of opioids used over 4 days in 4 groups of patients following a hemorrhoidectomy. And the 4 groups were low dose Exparel, 66 milligrams, middle, 199, 266, and then compared it to straight up bupivacaine at 75. The thing that I find interesting about this graph is over time, everybody needed more drug, and the people with the least amount of bupivacaine needed the most amount of drug, and the people with the most amount of bupivacaine needed the less drug. And I would ask you to rotate the dark black and put it second in each set of data for each time it was assessed. And it looks like a dose-response curve. Their conclusions were that the liposomal bupivacaine at 266, you see with the little stars there, was better. The issue here is one of perhaps choosing an experimental design that didn't really explore equivalent doses. Here's one that's a study that directly addresses the issue that I raised, that I encountered clinically. It comes to us from the Journal of the Knee, and it's a randomized, double-blind, dose-ranging study comparing wound infiltration of diprophone bupivacaine to regular bupivacaine for knee arthroplasty. Here's their methods. They're using escalating doses of Exparel up to 532 milligrams. That's double this dose here. And they compared it with bupivacaine 150 milligrams. This is an area under the curve metric of pain scores over 5 days. And they found that the higher dose right here did better than all of these doses over time. One of the things that's troubling about this graph is that they stack the results day on to day to day to day. Don't give an individual day by itself. And this is a graph that presents the different doses and the clinician perception of how well the pain control is going. And they found that with the 532 milligrams, it was somewhat better than the other doses. Here's the graph that I like the most. It shows the plasma concentrations. And even with the super high dose, 532 milligrams, it doesn't go anywhere near toxic levels. So that's good. What's bad is there's no metric of variability. Is there an outlier or what have you? The other thing that's interesting is if you look at the bupivacaine, straight up bupivacaine, 150 milligrams, and the liposomal bupivacaine, 133 milligrams, and the time course and the peak concentrations look very similar. Very similar. They concluded that the dose of Exparel of 532 milligrams was associated with statistically significantly greater analgesia compared to bupivacaine. Early in its marketing, they promoted Pacira, the company that markets Exparel, that it would last up to 72 hours. In September of 2014, the FDA issued a warning letter. And the warning letter said, specifically, there are new uses which lack approval and for which the labeling does not provide adequate direction for use. False or misleading because it overstates the efficacy of Exparel. And use of Exparel in surgical procedures other than those for which the drug has been shown to be safe and effective, and suggests that Exparel is more effective than it has been demonstrated. So that was in September. In April of 16 of this year, April 16th of this year, the Pacira acknowledged that it had received a subpoena from the federal prosecutors wanting more information about the Exparel marketing. Last month, Pacira issued a news release that said, Pacira Pharmaceuticals seeks court injunction to defend its right to share information about Exparel, consistent with its FDA-approved indication. So they're giving some pushback. And this, just October 16th, just a few days ago, from Law 360, Newswire Business for Lawyers, reports that the FDA has quietly unpublished a warning letter objecting to the promotional practices of Pacira. So it sounds like there's definitely some push and pull on this issue. between Pacira and the FDA. The focus that I would suggest that we take is to look at the data, and it has come to my attention that some new data are coming out looking at tap blocks and knee surgery that may show that Exparel does have a longer effect than bupivacaine. But in terms of what drives us clinically, I think we need to be grounded in the data as much as possible. Show us data, allow us to make informed decisions. Well, in summary, I would hope that for this particular drug, we get outcome data that compare liposomal bupivacaine to bupivacaine at near equipotent doses. Sounds like some of that may be coming out soon. And maybe we ought to measure plasma concentrations following administration of Exparel mixed with other local anesthetics, because that's what people are doing, and really determine whether it's safe or not. Well, this has been a lot of fun. Thank you for your attention this early in the morning, and we'll go on to the next speaker. Thank you very much.
Is this microphone? Hello? Yes, okay. So maybe we have time for one or two questions. I see you first. I'm going to here. Going to run around a bit.
Thanks. Rob Sneed from Plymouth, UK. I was very interested in the bupivacaine concentrations thing. We've been measuring bupivacaine concentrations in patients having multi-day catheter tap blocks for procedures like cystectomies and major abdominal surgery. And we've measured some spectacularly high concentrations and some highly heterogeneous concentrations. And as a result of that, in our district, locally, we've halved the amount of bupivacaine that goes into the tap block with seemingly no change at all in the efficacy. So there's a bit of a disconnect there. And I'm left with a very uncomfortable feeling that nobody really knows what the toxic doses of bupivacaine are. It probably matters a lot how quickly you got there and how long you stay there. And I think there's a whole pile of uncertainty sitting around there.
So you have another comment there.
Hi, Steve Schaefer from Stanford. The FDA commented on the bupivacaine levels in the Pacira review and noticed that some of the bupivacaine levels were so high as to be incompatible with life. And yet the patients actually were seemingly unaffected. And at least in the case of liposomal bupivacaine, my guess, and it's just speculation, is that the assay, is that the liposomes themselves got in the plasma where the bupivacaine was bound. And then the assay removed the bound bupivacaine, and that's why the drug did not have, the patients were alive, because the drug was in fact in the liposomes, but the assay didn't perceive that, which allowed you to have astronomical levels. I mean, levels that were 10-fold higher than where you expect cardiac toxicity, with the patients perfectly fine. And the only explanation I can come up with that was that actually it was floating liposomes that were picked up in the assay.
Thank goodness for the liposomes.
Any questions? No? So I would like to thank Dr. Johnson for his fine lecture. It's a good introduction of the day, and it gives some preview for what to expect in the session 2 on the innovation and entrepreneurship, because all these drugs need to go through some, or they need some entrepreneurship to get onto the market. So that's the topic for the rest of the day.